Welcome to Ajad Bioacademy. Today I will talk about second messenger. In pharmacology, in our cellular signaling pathway, second messenger is very much important. Within our cell, different types of organelles, different types of proteins, enzymes, factors are there. But some specific points, some molecules can be considered as a second messenger. Why these are called second messenger? Why not others? Okay, this is a very important question. Today I will just make you clear why it is called second messenger and what is the main role of second messenger. The topic is first messenger and second messenger. So actually our main focusing point is second messenger. Okay. So why it is called second messenger? Before going to the details, just I will explain in very shortly. That is, if you consider this is your cell, so most of the drugs and ligands are not allowed into the cell to show its activity, because within your cell has different types of organelles, and different organelles has different specific functions. So if all other ligands in endogenous or exogenous or drug can enter, then the cellular function will hamper. So most of the drugs or ligands are not allowed. Some drugs are allowed for lipophilic and low molecular weight. That can be hormones, vitamin D, steroids that can enter into the cell. That is the difference. Okay, so most of the drugs cannot enter. So how they will show their activity? They will bind with specific receptor over there. Then it will just send the message to the inside. Okay, and which molecule will just capture that message and it will amplify the particular point, that substance can be considered as a messenger. Not all the substances are not considered as a messenger. So here the main point is this one is first messenger. First messenger, as it bind with the receptor to show its activity. So which substance or ligands or drugs bind with the receptor, those substances or drugs or agents are considered as a first messenger. Okay, that can be drug, endogenous, exogenous or endogenous, hormones, neurotransmitters, autogoids, such type of substances, receptor oriented activity showing molecular first messenger. Different types of substances are present within our cell, but not all are second messenger. Why and what are the main characteristics to be a second messenger? Okay, so now come with an example. So suppose this is your blood vessel, and in your blood vessel, receptor, that is M3, muscarinic receptor is present, and this one is of course acetylcholine. Okay, so now what will happen? So over there it will just pass through the blood vessel and it will work to dilate the blood vessel. You know, acetylcholine is parasympathetic neurotransmitter, so it will dilate the blood vessel. How? When acetylcholine will pass through the blood vessel, you know, blood vessel has basically two areas to be discussed today. That is endothelial layer, inner layer, and made up of endothelial cell and smooth muscle. Okay, so now you consider the inner endothelium layer. So you can consider this is endothelial cell. Okay, and on the surface of the endothelial cell, this one is a M3 receptor. That means muscarinic receptor 3, and this one is acetylcholine. Okay, M3 receptor is actually G protein coupled receptor, and this G protein is GQ. Okay, GQ. So I will not go to the details about the G protein because we have a video in my lecture, so you can go for G protein coupled receptor. Okay. So what will happen when G protein will be activated? That means when acetylcholine bind with the receptor conformation will change, and it will just activate the G protein. That G protein activated G protein will just activate some enzymes, that is phospholipase C beta enzyme, and that enzyme function is to just change the PIP2 into IP3 and DAG, diacylglycerol. That means phospholipase C enzyme will act on PIP2, and PIP2 will turn into IP3, that is inositol triphosphate and diacylglycerol. Okay. Then what will happen? This IP3 will come to endoplasmic reticulum, which has protein modification activity, but one of the another function is to carry calcium. So you can say storekeeper of calcium. So these are calcium. Okay. Then what will happen? It will bind with the IP3 receptor, and the calcium will come out from endoplasmic reticulum. So before binding of IP3, the calcium were within the endoplasmic reticulum. Now it is in cytosol. So calcium level will increase. Then what will happen? That calcium will bind with the calmodulin. Then it will make calcium calmodulin complex. Okay. Then what will happen? This calcium calmodulin complex will activate an enzyme, that is ENOS, that is endothelial nitric oxide synthase. Then what will happen? This enzyme will just convert the L-arginine to nitric oxide. Okay. Then what will happen? This is gaseous nitric oxide, and it will just diffuse into the smooth muscle of the blood vessel. That means in muscular layer. Okay. So it will diffuse into the nitric oxide into the smooth muscle of the blood vessel, and here present soluble guanylate cyclase. Okay. Guanyl cyclase. Then what will happen? That guanyl cyclase will convert the GTP into cyclic GMP. Okay. And that cyclic GMP will activate PKG. That PKG will activate another one, that is myosin light chain phosphatase. Okay. So phosphatase means it will cause dephosphorylation. That enzyme will dephosphorylate. That means this is your myosin is already phosphorylated. Okay. And this is the myosin, and that will be phosphorylated. That means inorganic phosphate will out. Then what will happen? It will phosphorylated myosin will be simple myosin. That means single myosin. It is dephosphorylated myosin, and this single myosin is unable to bind with the actin, because in smooth muscle only phosphorylated myosin, that is active myosin, can bind with the actin. Then it can cause actin myosin complex, but actually the myosin is not phosphorylated, so it will not be possible. Okay, so no actin myosin complex, so no contraction, no contraction. Smooth muscle contraction is not possible because myosin is not phosphorylated. So now vasodilation. Okay, so why I am discussing the mechanism of acetylcholine? Here I will explain the second messenger very precisely and very elaborately. Okay, so now acetylcholine is the first messenger because it binds with the receptor. Okay, now I will just focus on some characteristics of the second messenger. First point is it should be non-protein. Some non-conventional second messenger can be considered as a small GT page, but it is actually not the mainstream second messenger. So you can easily say second messengers are basically non-protein. So they are low molecular weight. They will quickly produce or synthesize and very quickly they will degrade. They must have amplification activity. That means quickly it can change the activity of some substances, and they are very much specific in their action. That means they will work very specific manner on specific substances. Okay, they are very diffusible. It can diffuse within the cell very quickly because most of the second messengers are hydrophilic. Okay, so non-protein, diffusible, amplifying capacity, low molecular weight, it will work very quickly, degraded very quickly, that means half-life very low. How I will memorize and how I will choose this one is the first messenger, second messenger. So now come to the main point to understand very precisely. That is, your brother staying abroad will send a letter or will send money to your family. So who will send money or who will send letter? Your brother. So your brother is a first messenger, and depends on the sending letter or money, postman will come to your house. Without sending money, without sending letter, postman never come to your family. Within your family, several types of people are there. Your neighbors or your relatives may come in your house, very usual manner, but the postman will carry a letter or money sent by your brother. That would be the second messenger. Okay, so second messenger, that means postman, will come only based on your brother's letter or money. Okay, so now come to the point why I am trying to say like that. That is, if this is the first messenger, acetylcholine, it will bind with the receptor. So the postman will come only when needed. That means postman is not available within your house. So now here G protein were present but that was inactive. After acetylcholine binding, then it will be activated. Your PLC was present PIP2 was present, but this IP3 was not present. DAG was not present. DAG and IP3 come from PIP2 with the help of phospholipid C. Okay. Your calcium was present within the cell but not ready to work because calcium was present within the endoplasmic reticulum. But now this calcium is in cytosol. Okay. So that calcium is in a larger extent. Okay. Calmodulin was present. That was also, enos also present, but this nitric oxide was not present. This nitric oxide comes from arginine with the help of endothelial nitric oxide synthase. Okay. Now come here. GTP was present. Guanylate cycle is present, and cyclic GMP was not present because it comes from GTP. So this one is just newly synthesized. So now come here. So this is the first messenger. G protein is first messenger? No, because you can say this is your family members. Easily, very regular manner, they will transfer their internal message, but not external message. Okay, so this is your second messenger. Based on the first messenger, that will newly synthesize or produce, and they will follow all the characteristics. Non-protein in nature, they will amplify to release the calcium, and that calcium very quickly bind with the calmodulin and change the conformation. That means it has amplification activity, and it has the amplification activity on endoplasmic reticulum. If you say the DAG, it will activate the PKC, and that PKC will work on your nucleus, that is genetic material for gene expression. So that diacylglycerol will activate the PKC, and that will work on the further activity. Okay, so this diacylglycerol is the second messenger. Now calcium second messenger, nitric oxide is second messenger because nitric oxide is newly synthesized, very quickly synthesized from the arginine. Okay, and here cyclic GMP is second messenger, but all other molecules were present but inactive, and based on the activation they will work on another molecule. Okay, so just from this figure I just tried to make you clear that based on the first messenger, newly synthesized molecules can be considered as a second messenger, and they will follow the characteristics that in non-protein amplifying activity, very much specific in their action, and that will be diffusible, that will be low molecular weight, that will be very specific in their action. If you just try to connect those characters within IP3, DAG, calcium, nitric oxide, cyclic GMP, then you will get the answer. So those are considered as a second messenger. Other than G protein is not second messenger, your PLC is not second messenger, PK, PKC, calmodulin, enos, GTP, PKG, these are actin, myosin, these are not second messenger. Understand? So based on the first messenger, when some molecules will be synthesized very quickly, those are basically non-protein and mostly hydrophilic. Only exception you can say, this one is hydrophobic, that is lipophilic, and this one is gas. Other than all, that means cyclic GMP, calcium, IP3, these are the hydrophilic second messenger. So now very quickly another second messenger I will explain, then I will just list out all the second messenger and first messenger, then I will complete the lecture. Okay, so now cyclic AMP, another second messenger is missing here in this mechanism. Okay, so come to the that point. So another blood vessel within your respiratory tract, within your lungs. Blood vessel carry a receptor, that is beta 2. Suppose this one is receptor and this one is the adrenaline or noradrenaline. This is beta 2 receptor. Then what will happen? Now you can consider this is the membrane of your blood vessel of lungs, and it will carry a receptor. That receptor name is beta 2, and you know the with beta 2 it will bind noradrenaline, and beta 2 is actually the receptor is GS, that is stimulatory G protein. When noradrenaline and adrenaline bind with the beta 2, then will activate the GS, and that GS will activate the adrenalate cyclase. Then what will happen? With the help of adrenaline cyclase, the ATP will turn into cyclic AMP. Okay, that cyclic AMP will activate the pKa. That pKa will just activate myosin light chain kinase. What it will activate? How? No, it will not activate. It will just phosphorylate the myosin light chain kinase. In my previous explanation, I just wanted to say phosphorylated myosin, only phosphorylated myosin in smooth muscle can bind with the actin to make it contraction, and that myosin will be phosphorylated by the myosin light chain kinase. But here the pKa will phosphorylate the myosin light chain kinase and make it inactive. Okay, so it will not be able to make the myosin to myosin phosphate. It's not possible because free myosin light chain kinase is able to convert or able to phosphorylate the myosin, but as it is phosphorylated itself by pKa, then the myosin light chain kinase is not active to make myosin to be phosphorylated. Okay, so now what will happen? The phosphorylated myosin is very necessary to bind with the actin. So as it is not phosphorylated by that matter, so actually this is not phosphorylated myosin. Okay, so then it will not be able to bind with the actin. Okay, so no actin myosin complex. No actin myosin complex. So contraction, no contraction. Then that means vasodilation. Okay, so here same as before, just try to look back to the previous mechanism. Here G protein is not second messenger, ATP is not second messenger, adrenaline cyclase is not second messenger, pKa is not second messenger, myosin light chain kinase is not second messenger, myosin, actin, those are not second messenger. Here this is newly synthesized, come from the ATP by the help of adrenaline cyclase. So only this one is the second messenger. Okay, so this is the second messenger, and other things were already present, and they will show their activity or they will contribute the signaling pathway only by their activation, but they have no any amplification activity. They will just activate that one, that will activate that one, but the second messenger has the amplifying activity, quick modifying activity, and it very much specific for their action, and they will diffuse, has diffusible activity, and they are non-protein in nature basically, and other parameters already I said. Okay, so now very quickly I will enlist the first messenger and second messenger, and then I will just sum up my complete my lecture. Okay, so here this is the first messenger, and if you say this is the second messenger. So first messenger, you can consider your neurotransmitters, or the first messenger can work as first messenger. You can say hormones are first messenger. You can say water currents, first messenger. In that way, endogenous different types of substance which can bind with the receptor that can be considered as a first messenger. And you can say the drugs which basically works on receptor, those are first messenger. You can say alpha agonist, you can say acetylcholine muscarinic receptor agonist or antagonist. Those are that means drugs can first messenger. And what are the second messenger? Already I said, that means cyclic AMP, cyclic GMP, your IP3, you can say nitric oxide, you can say calcium, you can say DAG. Those are the mainstream second messenger. Okay, among them, these are basically hydrophilic, actually this one is gases. Okay, and this one is only exception, that is the hydrophobic, that is lipophilic. Okay, already I said most of the second messengers are hydrophilic because within your cell it is aqueous environment, so hydrophilic substance can easily diffuse. Okay, and all are non-protein in nature. Okay, already I said some second messenger, not conventional second messenger like small GTPase or other things can be a protein in nature, but these are non-protein in nature. These are small molecular weight. These can easily diffuse. They can work very quickly. This can be synthesized very quickly, and this can amplify the specific factor. So these are the second messenger. These are the first messenger. Okay, this is all about first messenger and second messenger. So thank you very much.